So it is the proverbial question of our time, right? The chicken or the egg. I'm going to geek out here and get in touch with my inner internet troll and tell you that it is most definitely not the chicken, but instead the egg, which evolved first, long before the modern chickens evolved. So today we're going to talk about the amniotes and what that means and the small little changes that happened in the egg that allowed for us to take over. But first, let's start with the amphibian egg. So the amphibian egg is basically the same type of egg that fish had. Amphibian eggs can be essentially described as an embryo and its yolk sac surrounded by a gelatinous membrane. So we call the embryo the animal pole, we call the yolk sac the vegetal pole, and the surrounding capsule, we call it the jelly capsule, go figure, and there's a vitelline membrane. And the vitelline membrane is basically there to facilitate gas exchange. It's a very permeable membrane that serves to allow the animal to exchange with its environment, its watery environment. The jelly capsule, on the other hand, is a substance that's designed to make it a little more dense, allowing it to sink into the water column or to become sticky and be able to attach to other eggs in mass. Now, the waste that this embryo produces in this amphibian egg is ammonia. Why is ammonia a good idea in this situation? Well, if you recall from our earlier lectures on fish, ammonia is a waste product that can easily diffuse through a membrane, and it's highly soluble in water. So fish use ammonia as their main excretory product because they can simply diffuse it out of their gills to avoid that buildup, which is toxic to physiology. So they do it too, because they're in an environment that's fully saturated with water, whether it's standing water or some other kind of micro environment where there is some sort of water present. So it's a very easy and straightforward and energetically cheap way to get rid of the buildup of nitrogen products in your blood, which could be a problem. And amphibians have taken this to the extremes that they could. So you're probably familiar with amphibian eggs in ponds. I know I get them in my swimming pool sometimes, but they could also be attached to the underside of leaves or on reeds in a special gelatinous casing that basically holds water, right? So a big water bag. And if you're something like this midwife toad, for example, you can even carry them on your back. She's called the midwife toad because she actually carries them all throughout their life cycle. So they'll actually turn into tadpoles and just stay on her back for the duration as she visits from pond to pond, right? And she does so because they're covered with water. So this worked well. And during the Carboniferous period, you saw the beginnings of Pangea forming in its entirety. And it had basically one ocean, the Panthalassa. And we call this not only the age of amphibians, but usually refer to it as the age of the swamp because of the sheer abundance of freshwater environments that were restricted all along the coast. But if you have a continent like this, that is this large, what do you think the climate's going to be like in the interior? Well, you probably guessed that the coast might be very temperate, being right by the water, swampy environments, hot, humid, kind of like Florida. But as you move towards the interior, you're getting further away from that moderating effect of the ocean, and now you're going to get into a drier and drier environment, right? And in many cases, it was essentially a desert. So during the Carboniferous is when you started seeing the tetrapods show up, and the tetrapods basically you started seeing these large amphibians. They're taking over all these swamplands. You're also at the same time seeing really large insects, really large arthropods. You're seeing the beginning of a lot of the flowering plant families in the Carboniferous swamps. So it's quite a time. Now, when the amniotes actually show up is actually a bit of a mystery. We know that they were amphibians that looked like reptiles, and we call these, for lack of a better term, reptilomorphs, right? They look like they resemble or they have the shape of a reptile. And we consider these to be the ancestors to the creatures that will eventually become the amniotes or the basal amniotes. So we're talking about sort of lower on the amniote phylogenetic tree. But when exactly this took place, we're not sure. And the reason is because there's about a 25-year gap in the fossil record. And we call this Roemer's gap, named after Alfred Roemer, who was an American paleontologist in the 60s, who first identified it. Now, these gaps in the fossil record are present in quite a few times in history, and basically just signifies that we had a lot of evidence of fish fossils before the gap, and then suddenly we have lots of fossils of amphibians, but there's a period of time where we don't have a lot of fossils, or any fossils for that matter, showing any kind of transition. So Roemer's gap is just one of many gaps that are out there, and there's a lot of debate about why this is the case. It could simply be that we haven't found the places where those fossils, where the conditions were there to fossilize. Perhaps the environment was going through quite a geochemical change that didn't facilitate fossilization. We don't know. But what we do know is that there is a very limited fossil record during that time. But as we start to search, I understand that there's a one site in Scotland that is starting to bear some fruit to fill in some of these missing gaps in the Roemer's gap, for example. But we do know that eventually you get amphibians, and somewhere along the line, the amniotes were there. But here's the thing. We talk about the amniotes, and we often associate it with the amniotic egg. But truth is, the amniotes as a group actually predated the amniotic egg. The amniotic egg came later, because there were already adaptations that were present that allowed animals to move away from the coast and further inland. And these were already present in the fossil record, even before the amniotic egg became present. So what might some of these adaptations be, for example? This lizard here might give you some hints, perhaps. So think of differences between amphibians and reptiles, for example. Well, one of the things we talked about last time was how amphibians don't have claws. They don't have keratinized structures. You already start to see keratinized structures in animals, even before you find any amniotic eggs. So keratinized skin, scales, claws, right? These sort of things. You're also seeing some other evidence of adaptations that are a little more specific, right? You're seeing larger digestive tracts, longer intestines and renal tubules, which are just kidneys basically, that are there to essentially absorb water, right? So you're seeing more adaptations for absorbing water. You're seeing lungs get larger, which means the animal will eventually get larger. You start seeing that the lateral line is kind of a thing in the past for these animals. So you're starting to see some of these sort of adaptations arise. But we always talk about eggs. So let's talk about eggs and where you find fossil eggs. Well, you can find them anywhere, really. They've been found everywhere with the exception of Antarctica. I read somewhere that Antarctica has a lot of ice and it's hard to find fossils there. Makes sense if we really think about it. But there's fossils that have been found of eggs, of entire nests, of many different species. And one thing I do want to shout out to is Roy Chapman Andrews, who was self-proclaimed adventurer. He was a director of the American Museum of Natural History, and he used to do these big trips into Mongolia and in other parts in China and even Russia. And his big paleontological discovery was finding eggs and nests that dated back to the late Triassic period, and these eggs were pretty large. So have any idea what have produced these eggs? Well, large eggs in nests from the Triassic period, a good guess would be dinosaurs. So these are actually dinosaur eggs, and more specifically hadrosaur eggs, but that will be in a later talk. Fun fact about Roy Chapman Andrews, he looks like he's a scout leader over here, but he's got a pistol. He was very much an adventurer. A lot of debate about whether he had a kill count even. But his sort of tough macho ways helped to inspire cinema and the whole wave of sort of adventure films like Alan Quatermain and things like that. These are films from like the 40s, 50s, and 60s. And eventually they found their way to inspiring George Lucas with one of his characters, not a Star Wars character, the other one, Indiana Jones. So Indiana Jones was thought to be ultimately derived from this personality right here, who apparently was larger than life. So these are fossilized eggs. And they fossilized because they were able to be fossilized because they were kind of hard. So they had a harder shell. So that suggested that these were amniotic eggs. So what's an amniotic egg? Well, an amniotic egg is named specifically for membranes, and one in particular, the amnion. And the amnion is shown here surrounding the embryo in this diagram, and it basically provides a watery environment for the developing embryo. Yolk sac was nothing new. There is also, this embryo is producing waste. So this waste is accumulating in the allantois, and the whole thing is enveloped in another layer, the chorion. So there's actually 4 layers. Now what this did was it provided a lot of good stuff to allow your egg to endure in difficult places. And over time, this also allowed your egg to get larger. And a larger egg means a larger offspring. And larger offspring means larger adults can form. And this is what you see. You see this trend going from fairly moderate-sized amphibians to really, really large animals, including ourselves. So these eggs can come in a variety of forms. Some of them are very, very rigid in structure. Some of them can be very flexible. Can you think of animals that have either of these? Well, you might think of a rigid shell. Think of a chicken egg, right? A chicken egg, a bird egg tends to be kind of hard, right? You have to tap it on the counter to even break it open, right? As opposed to, say, a flexible egg like you would see in a reptile. Think of a turtle egg, right? Sea turtle eggs, they're kind of soft and leathery, right? Both of them are considered amniotic eggs, just with a slightly different feel. So the thing to remember about eggs in general is they're not just sitting in their environment. There's a living thing in there, and it's undergoing all the same physiology that the adults are going to go through, right? So, it's undergoing gas exchange, it's having to depend on nutrients, and it's going to have to worry about things like water loss and stuff like that, right? So these membranes help to facilitate all that. So here's a quick matching question. Here are the 4 types of membranes that I mentioned, and here are some examples of what they're doing. So see if you can match each one. Okay, let's see. Let's start with number 1. Which structure surrounds the egg contents? Well, there's a lot of things in the egg between the embryo and all those other sacs. What's the thing that surrounds them all? Well, that's going to be the chorion. So that one on the bottom there. So the chorion is the membrane that sort of encases everything else. All right? And it's not necessarily connected to the shell, the outer shell, although that does develop in time. Number 2, which structure is involved with gas exchange? Well, it's an interesting one, right? Gas exchange involves bringing in oxygen and removing carbon dioxide. You don't want that to build up. And there's one structure that is involved, and that is the allantois. So the allantois is the structure that serves to essentially funnel waste products and also acts as the lungs for this animal, right? So the developing embryo is going to be producing waste products. That's making its way into the allantois. It's also the source of gas exchange. So number 3, which structure gives the embryo an aquatic environment? Well, this one should be pretty straightforward because this is basically what we're talking about, right? The amnion. The amnion is the membrane that most closely surrounds the embryo, and because it surrounds the embryo, because it's fluid filled, it essentially mimics the aquatic environment of, say, the embryo in an amphibian egg or a fish egg, right? So you're basically taking the water and surrounding the baby, so to speak. And finally, number 4, which structure is a pleiotropic characteristic? Well, the only thing that's left is the yolk sac. If you recall, we see the yolk sac in the amphibian egg. We see yolk sacs in fish eggs as well. It's the characteristic that's sort of the first one. So it's the pleiotropic structure. Development, but also in evolutionary time. So here again is a picture of what a fish egg looks like, or an amphibian egg, and then what a reptile or a bird egg would look like, and then finally sort of the mammal version of the amniotic egg, which is basically all happening in utero, right? With the exception of a couple of exceptions. You might know about the platypus. We'll get to that in the future. So again, you see the yolk sac in all 3 of these, but you only see the other characteristics, the other membranes in the reptiles, birds, and mammals, right? The allantois, which is a large structure in an egg, a smaller structure inside a placental mammal. You see the amnion becoming a more distinct thing in mammals. And you see the chorion, which is surrounding the egg contents and kind of keeps it separate from the actual outside shell of the egg in the reptiles and the birds. That chorion will eventually become the placenta that the placental mammals have, that forms the barrier between the female and her offspring, right, her developing offspring. So, again, a dynamic thing that's under selective pressure like anything else. So one thing that characterizes amniotes is their ability to move. You see this large shift in the capacity to undergo a journey. And this is a hallmark of amniote evolution. If you think about amphibians, amphibians are confined to water, so they could never really leave their swampland. They're chained to their swamplands. They're chained to their temporary wetlands or their pools or whatever sources of water. But now that you're able to take that watery environment with you to allow your developing offspring to endure, and even in the driest places, you can now start to colonize new landscapes. And as you do so, you see this rapid pace in evolution. And even now, when you compare reptiles to amphibians, even though reptiles evolved later, there's many more species of reptiles than there are amphibians. Many more species of birds than there are amphibians. Many more species of mammals than amphibians. Right? And this is because they are now no longer confined to just one type of really wet freshwater habitat. So here's an example of three species. There's a pangolin, which you might know as a highly endangered species, where there's a lot of concerns about wildlife trafficking right now with these guys and poachers. But you see it there. It's basically an anteater that's surrounded by these scales. You see here the legs of a typical bird. Insert your bird here. It could be a chicken, a turkey, whatever you want to call it, right? But you see the scales on the feet. And then there's a snake there, a rattlesnake, and you see the scales on it as well. All three of these animals have scales, and they evolve them independently and for different reasons. So what do we call a situation like this, where scales evolve independently in very distinct and different groups? One's a mammal, one's a bird, one's a reptile. Yeah, I'm sure you picked up on the convergent evolution that's happened, right? So all three are scales, all three come from the same source, but they've evolved independently for different purposes. So a classic example of convergent evolution, if I ever saw one. So if you're going to move around your environment, you need to develop those characteristics that are going to allow you to do so. So one thing you see is the forelimbs starting to develop. If you think of an amphibian, if you think about a frog, those forelimbs are small and reduced, and they don't have a lot of control over them, right? And the reason for that is that there is a specialized nerve plexus, which simply means like a net of nerves, called the brachial plexus. And the brachial plexus basically innervates your forelimbs and allows you to control all the various muscles on your forelimbs. When you look at amphibians, they only have 2 nerves that feed into this plexus. But when you look at the amniotes, you have at least 5, and in some cases upwards of about 10, that allow you to control all the different movements that you're engaged in with the front of your body, right? So this represents a major milestone in the ability to move through your environment, right? You're now able to not just rely on hopping or slow walking, you can now run. And if you think about it, there aren't many examples of amphibians that run very fast, right? Now you can run. So there's muscles that control your movements, and there's muscles that control your breathing. And obviously, the more you run and the more you move, the more you need to breathe and get that gas exchange going. The ancestral condition is to couple your movement with your ventilation. And by ventilation, we're talking about the movement of air in and out of your lungs. So if you think about the top figure here, which shows the left and right lung of a lizard, every time that lizard does kind of like, if you think about doing like Russian twists, for example, in the gym, you're twisting your torso, you're putting pressure on one side and freeing up space on the other side of your body. When as lizards walk, they're doing essentially the same thing. And they're basically inflating one lung at a time as a result, because those muscles on the edges of the body wall are pushing on the lungs. So for this reason, they can only take in so much oxygen. So whenever they're walking around, it's not a problem. But when they want to run, when they want to sprint, you might know that crocodilians, lizards, they're capable of very rapid sprints, but a lot of that's going to be anaerobic, meaning that they're going to rely on metabolism that doesn't have oxygen as a result. They also have their limbs out to the side, which doesn't help, right? That's part of the problem. So that's the ancestral condition. Eventually, you're going to take your limbs and you're going to move them underneath your body. So all of a sudden, you're not doing a side-to-side motion anymore. You're doing a front-to-back motion. Now, what that also means is you're now not allowing your body wall to push up against your lungs. Instead, now your lungs are going to inflate and deflate with your movement. And what's going to drive that is not going to be the muscles on your body wall and some special sort of locomotion / ventilatory muscles, but instead this new muscle that separates your thorax from your abdomen, known as your diaphragm. So your diaphragm becomes what we call in physiology the primary mover. So it's going to be the main muscle involved in that contraction, relaxation that causes your lungs to expand and contract. There's other muscles as well, like the muscles in your ribs, your intercostal muscles. So when you're in the full throng of a run, like this cheetah is, right, or a dog, as in the picture below the lizard, you see that the lungs are in the process of expansion. When the animal puts its limbs underneath it again, you're now in a contracted position. That's going to be your exhalation. So you're timing your ventilation with your movement. So the faster you run, the more oxygen you need, the more oxygen you get. The slower you run, the less oxygen you need, the less oxygen you get. It's awesome. And this allows for aerobic respiration to continue to power your movement, and allows animals to engage in very fast actions like this cheetah, this fast running action, or it allows them to do a sustained run like humans did, and wolves and other things like that, right? So it's just kind of interesting coupling locomotion with your ventilation, right? Now, in time, the amniotes will boil down to two camps, and I think it's safe to say that they're just two different ways of achieving the same results, which is surviving in your environment. No one is better than the other. And these two lineages will eventually form the diversity of vertebrates that people tend to think about on land. So you're going to see the evolution of the synapsid group, which will eventually become the mammals like us, and the sauropsid group, which will become the reptiles and the birds by extension. So these are two very different ways of doing things, and they've independently derived characteristics that they share. Can you think of some of these characteristics that they might share? Well, there's any number of things, and of course, whenever you're comparing a broad group like this, there's going to be exceptions to everything. But some things, for example, that are now characteristics in both groups, flight comes to mind. Mammals have bats, birds can fly, pterosaurs can fly. We'll talk about them later on. A lot of them go back to the water. You see the cetaceans going back to the water, right? Going back to the oceans and occupying river ecosystems and things like that, right? So things like whales and seals and stuff like that. You see the same thing happening with reptiles, right? You see animals going back in the water in the form of either turtles or, for example, crocodilians and things like that, right? So you're seeing basically the same sort of environments being taken over with slightly different modifications, right? And there's, of course, other things you can pick, right? Integument. Right, they get thicker skin. They start to develop scales in one group. That keratin becomes the hair in mammals eventually, or the feathers in birds. Right, so there's all sorts of things. They're just different. Now, the funny thing is, when you want to separate out synapsids and sauropsids, there's two totally different groups. They look totally different. I'm pretty sure you'd be able to tell which one falls into which group if you were to see a representative. But in the fossil record, what we use to characterize them out of all things are the holes in their head. Yes, of all the characters out there, that's how we define them. So, synapsids are characterized by having two what they call temporal fenestrations. Right? So these are holes that synapsids have one temporal fenestration. So that's one hole behind the eye. So they have their hole in the eye, and then behind the eye, they have one temporal fenestration named for the fact that it's happening on the temporal bone. As opposed to sauropsids, which can either have none, as in the case with turtles, or they can have two, in the case of birds and other reptiles that aren't turtles. And the anapsid, the no-hole version, and the diapsid, two-hole version, are both falling under the umbrella term of sauropsid. So it's thought that, and it kind of makes sense, that evolution probably favored having no holes behind the head in the temporal bone temporal fenestration. So that's probably the primitive amniotic condition, and turtles as well, although turtles, as you'll learn, are a very complicated thing in general. And then you see this split between those that had one and those that had two. Right? So these holes in the head, these temporal fenestration. Fenestration simply means hole, temporal simply means temporal bone. But what do you think these things are going to do? What do you think they're doing? What are they for? Why have these holes in the first place? Well, it's funny. Whenever these things happen, especially in the head, and there's holes, it can do one of two things. The cop-out answer is that it lightens the weight of the skull, which I've always thought was like, well, that's an easy answer. You could say that about any hole. Two, and more likely, it's going to serve as a place where you can attach muscles in a new way. Remember, bones are themselves nothing. What they are is an anchor for muscles to attach, and muscles allow you to do things. So by having a new surface exposed, such as in the case of a new hole, for example, that's a new place where you can attach muscles, or attach them in a different way, or attach them in a larger way to provide more force or something like that. So the one hole of the synapsids will eventually form into what's known as the zygomatic arch, which we'll learn a little bit later, right? So that's going to essentially be a big driver for mammal evolution, because we're going to attach a muscle there that controls our jaws. But I'm getting ahead of myself. So what are some of these other things going on? Well, another characteristic is they both breathe air, and both synapsids and sauropsids have lungs. And in both cases, you have the rib muscles that are helping to provide what we call the intercostals. These are helping to provide movement for ventilation. So when we talk about ventilation, we often talk about ventilation, we talk about respiration. Any idea on what the difference is between these two? The difference between ventilation and respiration. Well, respiration is the exchange of gases. So when gases move back and forth across a membrane, we talk about that being respiration. So when your lungs are taking in oxygen and pumping it into your bloodstream, that's respiration. When your cells are producing carbon dioxide and that gets in the bloodstream to get out of the body, that's respiration. So gas exchange means respiration. Ventilation simply means moving air. So just like you have a ventilation system in a building, same sort of thing. You're moving air in and out of a place. In this case, the lungs, tidal lungs, because they go in and they go out. So even though they both have tidal lungs, they're pretty different. In sauropsids, you see this faveolar lung or a septate lung, where you basically have a division, sort of like a canal that runs right through the lung, and then you have little pockets where gas exchange is going to happen, right? So the air is going to make it into those little pockets off of this central canal, and that's where the respiration part's going to happen. In contrast, synapsids have what we call an alveolar lung, where you have these branches that eventually terminate in these small little structures, alveoli, that are, surrounded by blood vessels where gas is going to exchange back and forth. So both of these, again, the goal here is to maximize that ability for respiration and to allow as much air to come in as possible. So the ancestral version was probably a very simple lung. So in amphibians, you have a very simple lung where there's just kind of like one membrane for exchanging air. But as you get bigger, you need more air, so you're going to evolve larger and larger lungs that need larger and larger surface areas with which respiration can work on. And finally, to accommodate all this, bigger lungs, you have to start changing your torso. Specifically, your neck gets longer, your head gets longer, you develop more of a trachea that's wider, allowing air to come in. So you're breathing in air, you're breathing in oxygen, you're taking that oxygen, you're putting it into your bloodstream. You're getting larger, so you have to do more and more of it. Well, the respiration system is one thing, and the other part is making sure that you're able to deliver that oxygen to all the parts of your body. So what you start seeing is also circulation changing. If you're a bigger animal that's engaged in more locomotion, well, now you have a lot more muscle activity, you need a lot more oxygen. So now to get that blood there, you need higher blood pressure. Problem with blood pressure is it can be dangerous if left unchecked, as we all know. And high blood pressure puts a stress on lungs. And if your lungs are especially delicate and thin, it can be a problem. So what you see is the eventual evolution of the two different circuits of blood, right? So fish started out with a single circuit, right? Blood comes into the heart, goes to the gills, oxygenated, goes out to the body, comes back in the heart. Simple loop. Over time, you start seeing that three-chambered heart in amphibians, right? And in reptiles as well, you start seeing that three-chambered heart start to separate in the middle, and specifically in the ventricles. And you see the beginning of a septum. A septum is just a division, right? It's just like something that divides something in two. So we call it an incomplete septum, which is probably what the first amniotes had, just kind of an amphibian heart, but with a little bit of a separation in the ventricle. So this incomplete septum is what reptiles, most reptiles anyway, still have. As time goes on, that separation is going to be complete, and you're going to transition from having a three-chambered heart and then having one that's a three-chambered heart with a small separation to finally a complete septum where you now can say you have a four-chambered heart. Now, the benefit of this is now you've separated your circulation out from simply having one loop to having two separate loops, a systemic circuit that feeds your body and a pulmonary circuit that's feeding your lungs and getting gas exchange happening there. So what ends up happening is you are now separating your oxygenated blood from your deoxygenated blood. And when you look at the birds and the mammals, you'll notice that the diagram there's no purple blood vessel. That's because the purple seems to be the universal color of mixed blood in terms of oxygen levels. So blue and red, no purple. So of these animals, which groups do you think will be able to maintain the highest blood pressure? Well, having the separation means you can now sort of control your blood pressure a little more, right? So now mammals get particularly large because they have a very high blood pressure, right? So mammals and birds as well, higher blood pressure because now they can separate their pulmonary circuit and their systemic circuit. Each of those is going to be powered by the same heart, so they're going to have really high blood pressure compared to some of these other groups, right? That have this mixing which causes issues, right? And that's why you see eventually the evolution of being warm-blooded happening here in the birds and in the mammals, because you need a certain amount of blood pressure to be able to do that. But again, getting ahead of myself. Another characteristic that you got to think about is, what do you do with all this waste you produce? Nitrogen, which comes from the metabolism of proteins and nucleic acids, when it breaks down and accumulates, can become very, very toxic, right? The natural course of events is for nitrogens to eventually become ammonias. Ammonia is great for being soluble in water, which means it can build up in the blood, but it's highly toxic, so your body has to get rid of it. If you're a fish, you leak it out your lungs, no problem. Amphibians leak it out your gills, or an amphibian leaks it out its gills into the water, no problem. But now you're on land, and you're an amniote. So you see less use of ammonia, and you see the beginning of the use of urea. Now urea is not as toxic. You could allow it to build up in your body. And it's thought that amniotes first started with urea. And synapsids, like ourselves, our kidneys still rely on urea, right? Urea is the main thing that's in urine, and our urine is liquid, right? Now we use our urine for more than just getting rid of nitrogenous waste. It also helps to regulate what's in our blood, but that's the way we do liquid urine. Sauropsids, on the other hand, their kidneys are not as efficient at concentrating, so they can't produce liquid urea per se. So instead what they do is they precipitate out acids, this crystalline acid, uric acid. And some of them also have specialized glands that allow them to secrete salt, especially saltwater species. So if you think about like marine iguanas, for example, sea turtles, seabirds, they often have these special glands that allow them to get rid of all the excess salt. But sauropsids, on the other hand, so we're talking about reptiles, birds, they're going to secrete instead uric acid, which is more of a solid form. So which of these is better for water conservation? Urea or uric acid? Well, you probably guessed. If you're producing urine all the time in liquid form, you're also losing water as well. Urea is just a small part of that urine. So you want to conserve water. Uric acid is a better way to go, and that's what you see. You see, dry desert habitats, for example. You'll often be dominated by lizards and things like that, and snakes, right? Sort of reptiles, as opposed to more tropical areas. All right, let's talk about senses. And people like to talk about senses because these new senses that allow them to move through their environment and understand what's going on will eventually drive the brain. So when you look at synapsids versus sauropsids, the ancestral condition is to have poor vision, to primarily rely on sort of other forms of sensation as opposed to sauropsids. Now, this is a bit of a loaded thing because we're basically comparing mammals with birds at this point. So, birds are known to have color vision. They usually have extra cones, right? So they have a lot of cones that allow them to see colors that we don't see, right? Whenever you look at a green thing or a blue thing, it only is green or blue because you're looking at it, right? But a bird could be looking at the same thing and seeing something totally different. We see white, and the reason we see white is because our eyes are saturated. But if you were to look at the same thing as a bird, you'd probably see all sorts of colors that we can't even comprehend or mimic, right? Simply because they're not saturated. They can see into these different realms of the spectrum that we can't. So sauropsids are loaded up with cones. They're allowing them to see colors. In synapsids, including mammals, generally rods are more important. Rods are more of the photoreceptor, again, involved in picking up light in general, and more of a sort of night vision thing. And we do have cones. 3 cones is what you'll get in humans, for example. So we see 3 in 3 particular ranges: blue, red, and sort of a greenish yellow. And those just happen to also be our primary colors, because guess what? Those are the 3 colors that we can actually see, and all the other colors that we can see are basically some mixture of that. If you were to ask a bird, they'd probably have more primary colors. Just saying. However, this lack in vision that mammals have is made up for in other senses. Olfaction, right? Olfaction, smell, bar none. Mammals are the best at the olfactory world, if you will, as compared to birds. Birds, of course, they're flying and stuff like that. They're relying on visual cues. Synapsids also have a much more adept hearing sense, and this is in part because of the middle ear, which we'll talk about the evolution of the middle ear from jaw bones, but that middle ear has 3 bones as opposed to the 1 bone in a sauropsid line. And then you're also relying on more chemical communication as opposed to the visual in the sauropsids. So in the end, you basically just have different lanes, and you stay in different lanes. So synapsids are going to be more the nocturnal groups, whereas the sauropsids are going to be more the diurnal group, right? If you think about lizards, they're out during the day. If you think about rats, they're out at night, right? If you think about birds, they're out during the day. If you think about bats, they're out at night, right? So they're taking up the same habitats, the same places, but they're using them at different times. Now, what's going to eventually happen is you're going to see that all these new senses require new brain power. And you see the evolution of 2 areas in particular. The forebrain, which is here indicated in purple, is going to develop and get larger, which is going to be a major player in understanding what this new sensory information is going to be. And then also the cerebellum, which is the part in the back of the brain shown here in brown, in this dark brown color, that's going to be involved in controlling movement. So you're going to see a more sophisticated animal able to better understand its world and to move through it. And just in time, too, because now the continents have joined up and they've become really large, and the environment's going to start to change. Those rainforests and swamps that dominated in the Carboniferous are eventually going to collapse. And this is important for us. Any ideas? Well, if you recall, it's called Carboniferous because that's where carbon comes from. The fossil fuels that we use are going to come from the remnants of these rainforests that become fossilized, and they show up as a layer, right? And if your country has that layer, then you've got that resource. So now you're getting Pangea completely forming, and this is the beginning of now the Permian period. So we're talking about 298 million years ago, right, is when it starts. So now you got the supercontinent. You're seeing a large interior, and you're seeing very little in the way of water. So the world's getting drier, and it's starting to show more of the continental climate where you're going to have winters and summers. Your summers are going to be unbearably hot. Your winters are going to be unbearably cold, and especially in the interior. You also have this large ocean that's going to drive these very, sort of monsoon events where you're going to get this sudden buildup in this regular rainy season that goes into the continent, right? And that's where the water is going to come from to feed the continent. And during the Permian period, you're going to see the beginning of some pretty incredible creatures that are terrifying at the same time. And here are two examples, Dimetrodon and Gorgonopsids. These two are going to come to dominate the Permian period, which is probably one of the most interesting times in the history of our planet. And the reason that they're going to be able to do this is because as the environment changes, there's going to be opportunities. And opportunities do not come without risk. And if you have risk, the only way that you can make it and make any progress is when you're willing to take that risk. So with that, I will see you when I see you with another video.